serve as kind of a roadblock to an enhancer from talking to a particular code. So if you have a sequence that you think is an enhancer, and you put it on the left side using your enhancer in RFP, you see that it blocks the enhancer from working on the left of the RFP, but it can still work on the GFP. Now, this is an experimental example, but this is observed in real life. Okay, so a lot of times in our chromosomes, we have insulated sequences that stop an enhancer from working on one gene but allow it to work. Another finding that's been noted is that in eukaryotic chromosomes, there are certain parts of chromosomes that have easy access to each other, and others that are kept separate. You can think of these as kind of loops. They call these topologically associated domains. So we'll go into more detail. But the way it works is that these insulator sequences that actually involve the protein cohesin, which we learned about also in chromosomes, the protein basically makes the chromatin on the left form one loop.
So I don't think there's sound.
Boy, this last couple of weeks have flown by. Here we are, last class. So we're going to finish up some discussion on cancer and give a very brief introduction to kind of population genetics and touch a little bit on human evolution. So we were talking about the cell cycle, and that's going to play an important role in cancer because in a lot of cancers we lose the ability to regulate cell division. So when we finished off last class, we were talking about the CDK cycle complex, and specifically the discovery of kind of the catalytic protein kinase. And so again, the CDK cycle complex is a heteromotomer protein consisting of a catalytic subunit called the CDK, which was originally identified in yeast as CDC28, and a regulatory subunit called the cycle. So the active form requires both the catalytic subunit and the cycle. And I mentioned before that the activities are going to be phosphorylating specific substrates, and phosphorylation is a modification that occurs on a protein that can either be inactivating or activating depending on the context. So the example I talked about was entering mitosis of the cell cycle, where you have to start to solubilize the lamins, and the phosphorylation leads. So how do we move around the cell cycle with a CDK cyclin complex? Because you need to do different jobs at each step. And what we're going to see is the catalytic subunit is there throughout the cell cycle, but it partners with different cyclins at different phases. So what we're going to see is that there's phase-specific expression of different cyclins, so you get activities that are different at each part of the cell cycle. And what's kind of really cool about it is the cyclin proteins are produced in phase-specific manners where they come on and off, and they each in fact help regulate the production and destruction of the new and previous one. So if you look at the production of cyclins relative to the cell cycle, you'd see that there is one specific cyclin, cyclin D, followed by E, followed by A.
I'm getting myself a drink.
You cannot get yourself a drink. I'm being so
What the fuck am I going for?
because you're not doing shit.
I am doing shit. I'm recording the video.
That's not doing shit. Slowly can fucking do that.
So you basically promote the cyclin and get rid of and that's both done on the transcriptional level and at the protein level.
So he's not taking
you can see that the name of the protein is called cyclin, which sounds like cyclin.
I'm being so serious.
And these proteins were originally identified in some marine animal species where they could study synchronized embryogenesis. And what they found is that they took synchronized embryos and looked at protein on gels at different time points. They see these proteins that come and go, and the appearance of the protein always corresponded to a particular phase of the cell cycle. So that's how they were originally. So now you can think about this that we've got the cyclins and decays that drive you through the cell cycle. They also allow the opportunity for regulation.
Why bother, bro? Because right after I say you always learn my evidence. That's saying.
Checkpoints are going to be there to make sure you completed the correct
test. You laughing because you know about to say that, aren't you?
You want to make sure you have a
Say you swear to God.
When you're about to leaving S and going
to you
want to make sure the DNA is paired correctly.
I just said it.
When you're getting to enter mitosis, you
Don't.
want to make sure that spindles are attached and prophase are done correctly. Now, you can mutate genes and pathways that block the checkpoints, and then the cell cycle just proceeds without any repair or anything happening. So in a lot of cancers, you're going to see damage to those checkpoints. So these checkpoints are a good thing. If you mutate the genes involved in the checkpoint, then you're going to be able to proceed without the corrections to chromosomal structure, corrections to DNA repair. So an example of a particular checkpoint here, we're going to look at going from G1 to S phase, which is called the RB protein, or retinoblastoma. So we're going to work through this here on the slide here to show you what's going to happen. Okay? Is that so let's focus on the event that we want to happen, which is down here, and then we'll talk how it's regulated. So when we go in from G1 into S, what we want to do is we want to degrade cyclin D and synthesize cyclin A, and that's going to allow us to go into S phase. How do we regulate that? Well, it turns out that there's a protein called RB, which stands for retinoblastoma. We'll see how that's involved later. And what happens is that RB is bound to a transcription factor called E2F. So when you're not wanting to divide, you keep RB bound to E2F, okay? And the cell stays arrested in G1. However, when you get a growth signal, say go divide, a CDK cyclin complex, CDK4 cyclin G or CDK2 cyclin E, phosphorylate RB, that causes RB to come off E2F, and then E2F can do its job as a transcription activator and turn on the genes that start cyclin A production and lead to cyclin D regulation. So this is kind of the sensing area. So if you want to keep yourself in G1, you don't phosphorylate RB. Once you get the signal to go ahead, you phosphorylate RB. So imagine that you have a mutation in the RB gene where it's knocked out and not functioning. You've lost this checkpoint. So now E2F is always active, and you can start S phase without getting inputs. Now this suggests that RB is going to be sensitive to some kind of regulation. Well, it turns out that there's an upstream regulatory pathway that's mediated by a protein called P53. P53, right, when active, right, makes a protein called P21, which actually blocks CDK4 cyclin D. So therefore, the CDK4 cyclin D activity is off, RB stays on E2F, and cell division doesn't occur. This should not really be an arrow here. I think this should be like a inhibiting sign, because P21's job is to inhibit. So you can take this information and put it back onto the slide here, and you can see that, okay, well, P53 regulates P21, which inhibits the CDK complex. And if the CDK complex is inhibited, you don't get phosphorylation of RB. It's a long set of cascades, but what you can pick up here is that if I knock out RB, if I knock out P53, or if I knock out P21, and knocking out means making mutant, so it doesn't work, cell division will be controlled. So mutations in any of those three genes, homozygous mutation, knockout mutations, in P53, P21, or RB, have all been associated with cancer. So those are important genes to always have. We also see that one of the signals that actually triggers P53 production is DNA damage and radiation. Because when would you want to delay your cell division? Before replication, right? If you picked up damage like diamine dimers, you would like to try to repair those. Because if you remember back to the chapter on DNA replication and mutagenesis, that if you could repair
I'm not getting what?
DNA's replication,
Because you're too much of a pussy to just pick up the other bro, Asia.
so you basically want to give yourself time.
Asia. You were just too
Now you get an
big.
extreme amount of
I
DNA
hope you
damage.
get food poisoning.
P53 can instead turn on apoptosis pathways, what's called programmed cell death. And the idea is that this cell has sustained so much damage that it's going to make lots of mutant genes. So we evolved to have this cell death process that just kills that cell off. It's better to lose a cell than to have a mutant cell. So this then introduces the idea of genes that, when mutant, lead to uncontrolled death. Okay, we talked a little bit about regulation of cell division, but also bring in signaling pathways, hormone pathways that turn on cell. So we're going to think of two kinds of mutants associated with cancer. Those that are hyperactive versions of cell division promoting activities, these are going to be dominant mutations that mutate genes that we now call oncogenes. So their normal role, as what's called a proto-oncogene, is to activate cell division in a controlled way when the hormone is present at a particular phase of development. However, you can make mutations in these genes that cause the protein to be constitutively active, so it doesn't require a growth signal. We saw that with RAS protein. Okay, so the oncogenes are gain of functions in growth promoting activities. They do a lot more growth promoting than the other class of mutations are loss of function alleles of proteins whose job is to normally keep the cell cycle in check. Okay? They're called tumor suppressor genes because their normal role is to slow down cell. So RB, P53, P50, P21. So if you have homozygous loss of a tumor suppressor gene, you've lost that break, and then cell division can occur. So you can think of oncogenes as kind of accelerators or gas pedals, okay? And you want to not press them too hard, but the mutant ones press too hard. And you can think of the tumor suppressor genes as brakes. So if you all of a sudden damage your brakes, you can't stop you. In most, if virtually all cancers and tumors, you have combinations of oncogene mutants and loss of function of tumor suppressor genes. Okay? So this picture here, this kind of shows you the same information. So here that you only need one mutant copy of the oncogene, okay, because it's a dominant allele. And remember, in its normal form, it's called a proto-oncogene. In its mutant form, it's called oncogene. The tumor suppressor genes, you have to have mutations in both copies, right? And so when you get the mutant allele, that's when you have lost that kind of control. So here's an example of a mutant form of an oncogene, that's a mutation in a proto-oncogene. This is the RAS pathway, growth factor receptor. So normally this only promotes proliferation when the growth factor is present. However, in the cancer cell, it promotes proliferation without having a ligand program. It's a very specific, there's some very well-known specific mutations that make the RAS protein be active by itself. And this is shown up here. Another example is kind of another way to get receptors that are hyperactive is just to make lots of copies of them. Okay? This is just showing with the ATR2 receptor, okay, which is normally promoting growth, okay, in a signaled way. When it becomes hyperactive, it can become hyperactive by having gene amplification, which means multiple copies. And the little colored dots here are showing lots of copies of the gene. Now, oncogenes were originally identified through a cell transformation assay. So remember we said that normal cells grow in a monolayer. So here's our normal mouse cells. They grow in a monolayer. These are not cancer cells. However, what they would do is they would take DNA isolated from a human tumor cell and transform that into the mouse cell. And then they would look for fast-growing colonies of cells that piled up on each other. You can see here. Remember we talked about the loss of contact inhibition. And what they found is that these transformed cells, as they were called, were due to taking up the mutant oncogene from the human DNA. So the human DNA, that's a cancer-causing mutation, the oncogene got taken up by the non-cancerous mouse cells and it made them disease. And this helped them identify the first oncogene substance. That remember your cell biology textbook? Jeff Cooper was one of the discoverers of this. Now, we talked about tumor suppressor genes having to lose both copies. There's kind of an interesting, genetic explanation for what we see with retinoblastoma. So retinoblastoma is one of the few kinds of cancer that's caused primarily by loss of function of both copies of the RBG. So in the cells that form the retinoblastoma, which is cancer in the eye, both copies are mutant. Yet when we look at a pedigree, you will see that even individuals who are genetically heterozygote will develop retinoblastoma at some point. So at the pedigree level, okay, at the whole individual level, we say that RB's inheritance, the mutant inheritance pattern is dominant. Okay? But at the cellular level, it's actually recessive to wild type. You have to knock out both copies. So how can it be both? Okay? Well, what researchers figured out was that there's a very high likelihood that by random chance, so there's no cell that you should almost every individual heterozygous for RB will develop retinoblastoma. But at the cellular level, what happens is actually very powerful. You were born homozygous for retinoblastoma, RB undefined. This is just showing
You just told me? What else is there now? The king's recording. What do you want me to do? You don't care, so what do you want me to do? Shouldn't go. On what? On what? What did I saw? I'm going to study it. Okay. I'm going to watch the episode. I'm going to watch the video on playing poker.
Watch on your phone.
How am I studying it? My computer's recording on the lecture right now. It's on 175 speed. It's done in 2013.
It's on Sony's. What are you even talking about? Shut the first of all, stop talking like that, bro. How would I know how to take it off?
would allow it to be
very different
and the other is to express or not express.
when you have to observe, you don't have to type.
So the end result is that we end up with different expressions.
Thinking about our little
We are either
data,
two
but then you pick
offering
type.
you or idea on two.
So if you were studying this, you would say, okay, there's something going on, right?
This
There's
was the same
my
regulation pattern is not happening. A lot of research
and you might look at this and say, hey, we have a right.
like
And
class.
it's got a certain meaning, right? Where you actually see higher level.
Allow idea two to be expressed locally.
This can be as simple as big A, big A, big A,
Allow idea one to be expressed locally.
now, what's interesting is that animating will bring the population back to the beginning. So let's say you started a population with armies with this, and one generation you have to and kind of a more biggest reason why we're studying theoretical knowledge is to actually understand biology, what's happening. Some of us want to why we're going to look at a couple of abstract terms. So, if you look at human populations, you see that there are human populations that their UI phenotype is behind other populations. And if you look at their psychology, you can see that there's real sequence of differences. So population goodness so with those lots of different charts, you can kind of see that the US UI, the receptor means that the different fields so while we have this particular population, what one of the factors that has to do with variety of population is that any time a population is spread, only a few and they bring with them whatever it is they have and establish resistance. I've also told you that sometimes with small populations, this is a lot more. So this is a computer model where we're going to get 30 generations, and we're going to only start with so you would think, okay, I've got wild type and this color line is just another number. And every starting population is 50%. One of the big but just by limited numbers of populations, what you see is that the real frequencies exist. It's very rapid. And many times you can see that one allele. Now, look what it was, because we only have midward. So this is sometimes normal, very limited trend, because it's a population, limited so that's a very small now, if we had a large population, say, we can see that the modeling, people live in state early, just like around 10 to like 100 people create deep learning make. This slide here, kind of see where it's related to this. Now, what happens as population moves? When that population may establish a new life. So if there's a large population, people in the numbers can readily and so it's some of the original population found new island west states that have population happened at the military because readily and legally nobody yellow. So this new population notice is lower. Right there. All of a sudden, this will be much bigger mix of blue. So this is what's on the bottleneck. we have a serious recessive wheel with a homozygous recessive X selection against the equilibrium. So we'll see that the equilibrium drop down to almost zero, but the heterozygous recessive will not have zero drop. Now, to kind of show that effect from high pressure, we're going to do a simulation where we start with a population that has a predominant of the less good wild types. We're going to see that over time, right, the more good allele is going down. So these are full time. And we start with 499 lines that are building, only one is the elite. So that means out of 1,000 alleles we start with. However, the homozygous will be has a fixed 0.98 compared to the non-homozygous. So if we run this simulation, we see that the allele hit rate essentially always a couple of other things that we so now we see if the alleles present in the population, one of the reasons why that is they are population. So the present so because that selection and rate factor is not necessarily and we don't see genes that are involved. So if we it's kind of a breakthrough. And what you can see is that after the population, we see starting of this is the skin wheel. We're going to first find out the population. We're going to look at some biological features first of all about the population. And what we can see is that all of those other areas and we see that there's a theory to say, okay, well, when the population is Africa, now at the top of that population, now we know now there's huge reduction in population size like so what is when you think of other individual players we see that there's a really different society with these multiple. So here we see again that there's a blue wheel for the population. But then here SLC is away. Europeans have a yellow wheel. And if you think about it, blue wheel. And then the ones and two are as a first color. How do you one so we see that the Europeans have the same wheel. So that has come that way. Now this doesn't tell you anything. Something that you know a little more about on X is where Larry. So we talked about Cyprilia. So kind of a comparison between so we see a high degree of how much Cyprilia with malaria. And so and we're going to base this on the idea that so the primary when malaria is high, Cyprilia is so we need to get this everywhere that when malaria is present, the heterozygote has to be one. Think about and the homozygous is the so when you have the selection of malaria involved, that is however, the non-malaria present population then the video what you see is the fitness that's also on this slide here we look at but any of you heard we talked about how the pesticide they need better on those sites with a basic pesticide and so now we're not going to be any of that stuff but we're looking at resistance so the pesticide on this video and we're going to look at the impact so we're going to start with the kind of the original you see that the homozygous resistance you don't really have it so if you let this big R had the highest one. It's the same thing like you. This is big R, so you're not going to do it like 12. The way it works is like, so sometimes CDT select, you basically, I see the T1 that you do that has business. So apparently that means like, this is true, and then little r, so that happens. So what does that tell you about the difference between little r and you know, onto the so there was an advantage of the left form. You see, it was rooted and then used kind of similar in a sense, wasn't right? Malaria population of malaria because they selected that located at that area. How do we talk a little bit about when we use that radioactive so that just finish up a couple of times. When we look at genetic so you took a wheel that says it's not single radioactive and so this particular example here is looking at one generation children tell how much where the great grandparents used to take radioactive and use to draw their blood particularly so the colors here represent wheel 1 we also get side differences. So over here, we see that the package represents. So if you've got something so your ancestry, that's what I'm saying. So it's like you got a top that's one is take some of those subset of people who do it. So when human populations are conflict, you see that there's much less diversity.
So better for speed, better for accuracy.
And this is really all the species in a lot of problems. So the difference in other so research in general, and each of us have some kind of less
tools that we can laboriously build ourselves, self-interesting problems. We've been 3 or 4 lectures now building groundwork, building foundational work. Give us a framework in order to think about today's topic. Building models. So building models is probably the most useful, widely used machine learning. The basic thing about not just natural science, but mathematics, computer science, and statistics, and especially social sciences, economics, ecology, and so forth. Building models. So what we'll do today is we'll talk about building models. To start talking about them, we'll run this down today. The rest came from Francis Galton. So Francis Galton was one of the sort of brilliant folks who lived in the 18th century. He was a cousin of Charles Darwin. He often hung out at Darwin's house and spent a lot of time talking about various things. Galton did huge contributions across all kinds of science. He really started with the idea of meteorology and weather. He had all kinds of contributions to exploration of the world. He was one of those sort of gentlemen explorers. And one of the things that he did a lot of was he was interested in measuring. And so this sort of started when his cousin Charles Darwin published Origin of Species. And Galton immediately picked up and started thinking about how what Darwin was saying about heredity, inheritance of characteristics. Like people, so he felt was interested in how heritable are traits. So he looked at the heights of parents and compared them to children. And he was finished with this chart that shows the average height of parents. How far they are above the mean, that's the average all across the continent. So across the continent, we studied people on average who were about 6 feet, 4 inches.
I hope you know you're just not gonna get any answers if you do.